In today's episode, we'll be going through enzyme exam questions. The way I'm going to do this is that I'm going to show you from start to finish how I'm thinking, approaching, and answering the questions. And remember that the mark scheme is written in the description box below. So let's begin. To which genus does this fungus belong? I know that I'm just going to look at the italic words, and so the answer is going to be Amanita. Next question says, what does this pattern indicate about the action of the fungal enzymes? Do you know what the pattern is? Because I don't. So that's why we need to actually read what's actually going on. So it says, the region of yellow staining shown in the figure includes part of the agar where the fungus had not yet grown. And then we have the figure as well. So, you think to yourself, well, it's enzymes. It can't be anything complicated. And although it's kind of like an application question, you can get away with just saying, extracellular or secreted, because that's how fungal enzymes work, by extracellular secretion, right? There's other things you can say by looking at the diagram, that the enzymes are released or diffused away from the fungus. Next one says, what can be concluded from figure 4 about the molecules or ions that affect the functioning of the nitrogenase enzyme? I love this question. It's sort of like an application question because you don't really know what the nitrogenase enzyme is, but they have given you the figure and the overall equation. So if they've given that sort of information, you have to use it. Plus, it's a conclude. So by just kind of stating what you see, you're going to get the marks. So let's have a look. What is going on? We can see that carbon monoxide, CO, is binding to the allosteric site. Now from your revision, you should know that allosteric site basically means, a non-competitive inhibitor. We can also see a cluster of those elements. Now if we have elements in like a circle represented, then that means it's a prosthetic group. We can also see that hydrogen can bind instead of nitrogen. That refers to a competitive inhibitor because they're competing for the same position of the active site. We can see that ATP is being used. So if a reaction is using ATP, does that not mean that that energy is required for the reaction? It does. So all those points that I've mentioned, we can write down as it's a conclude, and they don't really often explain sort of answer. Next one says, suggest 2 ways in which leg hemoglobin improves the performance of the nitrogenase enzyme. Do you know what leg hemoglobin is? Because I don't. So let's read the information. Leg hemoglobin is a molecule that improves the performance of nitrogenase. It has very similar properties to mammalian hemoglobin. Bingo. Whenever the exam question says it has very similar properties to X, Y, or Z, your answer is based on those properties of X, Y, or Z. And it says 2 ways. So we'll think about hemoglobin, about its properties, how it transports oxygen for respiration, and of course respiration produces ATP. We can also think about what actually removes excess oxygen, creates anaerobic conditions. Another thing, as I'm thinking about the carbon monoxide in the figure on the page before, that hemoglobin actually removes carbon monoxide to prevent inhibition. So those are 3 points, but we can just say 2. So that's the end of the video. Remember, the 2 most important exam techniques are read the question first and state all the obvious points. Now over to you. Try some questions and let me know how it goes.